have to develop as you go along is that things that are labeled as impossible, for instance, at one point in time, it was definitely impossible to clone a mammal. You could never clone a mammal, we could only clone frogs. So that was obviously down. And so Corrado is going to talk to us today about the big picture of some of his work. He has shared with me that he likes to work on sperm. He's interested in chromatin remodeling, and sperm is a good model because it doesn't divide. So you don't have to complicate your research with the cell cycle problem. So, Corrado.
So the title of my presentation reflects the work that we have carried on in Rome in the last 20 years or so, and is essentially focused on the role of epidermal spermatozoa as mediator of long-distance flow of information connecting the soma to the germline, and also as a second role that is the transgenerational vectors of such information to the progeny. So as I said, long distance soma to germline and transgeneration inheritance. There are evidence that this can happen. There is the work that Oliver Randall mentioned briefly today, and in which the recombinase expression in the brain was generated Cre-dependent effect in the epididymis. So long distance, meaning that the place where the experimental target was generated have the site of that were targeted were at a distance. There are other evidence of that. I like to remember the work of the Australian group of P. Berry, that showed that the microRNA delivered in the brain can be transported through the bloodstream to the germline and further delivered to the offspring. And this is our own work, in which we show that HSP and messenger RNA produced by cancer cells that were inoculated in male mice can be transferred via exosome in the bloodstream, reach the epididymis, and be taken up by the sperm. And I would like to focus on this a little bit more in details. So we use the melanoma cell that stably expressed the HFP. This cell were inoculated, subcutaneously inoculated in the limb of male mice. And about 40-45 days after inoculation, when the tumor was visible, we sacrificed the animal. We purified the vesicle circulating in the blood, and we extract RNA from this vesicle. And at the same time, we extract RNA from the sperm of the same animals. We screen this RNA looking for HFP RNA. And to make the long story short, we found HFP RNA in the vesicle of inoculated animals, but not in the vesicle, the circulating vesicle of non-inoculated animals. And similarly, we found RNA, HFP RNA in the sperm of inoculated animals, but not in the sperm of non-inoculated animals. We obtained an F1 progeny from these founders, and this is the screening of a litter of 12 animals. And in at least in 9 out of 12, in at least one tissue, the messenger RNA, HFP RNA was present. More in details, you see the animal 1 and 2 are negative. The number 3 is positive in the ovary and in the lung. This is ovary again and spleen, and here ovary and brain. spleen and brain. So it looks as spleen and brain are the most targeted tissue in this kind of experiment. So this really suggests altogether that there is a flow of RNA connecting the soma to the germline, and this is further delivered to the offspring and next generation. And this inspired us this model in which we represented here the tissue or the organs, especially those that are on the stressing condition, release RNA-containing vesicle in the bloodstream. This vesicle cross the vascular barrier, reach the epididymis, are taken up by the epididymal spermatozoa, and when the spermatozoa fertilize the ovocyte, then these are delivered to the ovocyte. They are further propagated throughout the development, embryonic development, and they are mosaic inherited in the adult individual. Now, from this picture, it emerged that a central role in the whole mechanism is played by the fact that the spermatozoa are permeable, epididymal spermatozoa are permissive to foreign molecules. And of course, this is it's important because if the sperm would not be permeable, then this flow of information could not reach the gametes, and of course, the gametes would not be able to deliver the somatic information to the progeny. So the permeability of the sperm, we studied this. We started studying this permeability many years ago. This is a quite old slide showing the interaction between an epididymal mouse spermatozoa with a fluorescently labeled DNA, and you see that the DNA is binding on the subachromosomal segment of the sperm head. More recently, we found that also a vesicle can be taken up and internalized. This is the interaction between the spermatozoa and rhodamin-labeled human exosomes. And again, you see there is an accumulation in the subachromosomal segment of the sperm head. Originally, we thought that this ability of the sperm to take up foreign molecules could be and to deliver them to the ovocyte could be used as a tool for generating transgenic animals. And according to a process that we call sperm-mediated gene transfer, in which the first step is interaction between sperm and DNA in vitro, and then the complex sperm DNA is used in IVF assays, and then the two-cell stage embryo is implanted into foster mother. And a proportion of the animal that were generated contained the DNA that was originally incubated with the sperm. However, to our disappointment, none of this animal was transgenic. And the reason is that the sequence we thought they were the transgene, they were not transgene because they remain as non-integrated episomal structure. So the animal had this foreign DNA, but the foreign DNA was not integrated in the host genome. So they were forming quite strange patterns that were unstable over time. However, this sequence can be transmitted from the founders to the F1 generation, and they were also transcriptionally competent. And at the time where this results were obtained, there was no obvious explanation for them. We also found that not only DNA can be taken up, but also the RNA can be taken up. And actually, this was an important turning point of our work, because the RNA allow us to discover that the epididymal spermatozoa are endowed with an endogenous reverse transcriptase activity. And this was observed using the RNA transcribed from a construct that contained a beta-gal reporter gene. So this RNA was incubated with the sperm, and then we extract DNA after incubation, and we simply amplify using a pair of beta-gal specific oligos. And as you can see here, we found a beta-gal cDNA in the sperm, and when this this complex were used for in IVF assays, we also found the cDNA in the 2 and the 4 cell stage. So it was transferred from the sperm to the early embryonic stage. We generate animals, founders with the system, and from the founders we obtain the F1 animals, and then we screen these animals looking for this beta-gal cDNA copies. And we found that they are mosaic distributed in the tissue of founder animals. They are always maintained at the low copy numbers, so below 1 copy per genome. They can be transferred from the founders to the F1 progeny. And again, in the tissue of positive animals, they are maintained as a mosaic and always at the low copy number. And so altogether, these results show that these sequences were not integrated and remain as extrachromosomal structures, and they are propagated from one generation to the next as a non-integrated extrachromosomal structure. However, they are transcriptionally competent. We found a mosaic expression, HFP expression in different tissue of animal of both populations, and this is an example. You know that tissue expressing HFP can be easily detected by a staining procedure, and they become blue stained. And you see there are some of them are quite heavily stained, and some others, so there is different level of expression, and those are control from the same tissue, but from wild type animals, where of course processed in the same way. And we call this sperm-mediated reverse gene transfer. Now, it is known that most of the reverse transcriptase present in the cells is encoded by a line 1 retroelement that is constituted by an open reading frame 1 that is coding for an RNA binding protein, and by an ORF2 that is coding for the endonuclease and reverse transcriptase activity. So in order to learn more about the reverse transcription step occurring in the sperm, we used the so-called retrotransposition cassette that is constituted by an HFP reported gene under a CMV promoter interrupted by an intron in the opposite orientation. So to make the long story short, to have this reported gene expressed, this vector should be first transcribed, the primary transcript should be spliced, and the splice RNA should be reverse transcribed. So what we did, we incubated the spermatozoa with increasing amount of this construct, 550 and 500 nanograms per million spermatozoa. The incubation was about an hour. Then we spun down the cell and we extract the supernatant, and we extract the DNA from the supernatant and from the pellet. And then we simply analyze this DNA by a direct PCR using a pair of oligos that flank the intron sequence, and that can allow to discriminate between the original DNA, that the original construct that generate the fragment, amplification fragment of 1,200 base pairs, while the splice form is characterized by a fragment of 342 base pairs. So the results are here. Those are the DNA samples from the supernatant and from the nuclei. And as you can see, we found, as expected, everywhere the large amplification. But when the sperm were incubated with the highest DNA concentration, also the splice form was generated, and most of it was released from the sperm and was accumulated in the supernatant. And only a minor proportion was maintained in the sperm. This is also a time-dependent process. and it's quite a quick process. 15 minutes of incubation are enough to make visible, to synthesize a small proportion of the splice form, and after 30 minutes, it's already quantitatively present. We use this complex sperm DNA to fertilize the egg, and we follow the fate of this sequence at different stage of development. And we found the splice form, but sometimes also the non-splice form in the two-cell stage embryo, in the fore, in the morula, in the blastocysts, in the fetuses, and finally this sequence were also inherited in the tail tissue of born animals. So these are not transient structures, they are quite stable, they survive throughout the development, and they are mosaic inherited in the tissue of born animals. There are also transcriptionally competent. We found mosaic expression always of GLP in different tissue, and this is an immunohistochemistry showing the expression of the GLP in liver. Liver, it's a high expressing tissue, and at a high magnification, you can appreciate that just few hepatocytes are positive. Most of the expression is in the vascular epithelium. And the same as in kidney, there are a few glomeruli positive, and most, again, is in the vascular epithelium, and also in the brain. It's a poorly expressing tissue. The little expression is in the vascular epithelium. So now the question is this, beside this single gene or simple traits, could also the complex multifactorial information could also be transmitted through this soma to germline flow. And we heard already, so I don't want to invest much time on this, that the epididymal spermatozoa have a cargo of small regulatory RNA, microRNA, circular RNA, dsRNA. These are essentially from somatic origin, and they are acquired by the sperm during the maturation in the epididymis. Now, most important is that the composition of this RNA, of this cargo RNA, it's highly dependent from the environmental factors. So the environmental factors to which the sperm donor is exposed has an impact on the composition of the sperm, of the RNA, excuse me. So this RNA cargo is delivered to ovocyte at fertilization and has the potential to affect the epigenetic and the metabolic of the developing organism, inducing alterations or disease like metabolic syndrome, diabetes, heart disease, cancer, and also producing sometimes the stress-induced traumatic behavioral changes. And this effect can, in some cases, can be stable across several generations. Now, the other important question is how can a regulatory RNA-based information delivered by the spermatozoa generate new traits. So now I have moving to the more speculative part of my talk, suggesting that the Waddington landscape metaphor could provide some conceptual help to answer this question. And for those that are not familiar, I will recall that Waddington is the first who coined the term epigenetic landscape, in which he described how a totipotent undifferentiated cell can generate multiple differentiated cells according to a differentiation process that he represented as a canalized pathway, as you can see here, in an epigenetic landscape constituted by hills and valley. So we were impressed about that. We were this model inspired us. And this is how we imagine the epigenetic, the remodeling of the epigenetic landscape of the embryonic epigenetic landscape induced by the regulatory RNA delivered by the sperm, that is represented here as this green ball rolling down from the top of the hill, that represent the epididymal, the landscape, the embryonic landscape. So we think that when this RNA, the regulatory RNA delivered by the sperm, can remodel the epigenetic landscape, digging this canalized pathway, this deeper canal, and at the same time silencing or inactivating, functionally inactivating the previous one, which are represented by this shallow canal. So now the formation of canalized pathway correspond to the formation of genomic circuit, coding circuit, that of course have the potential to remodulate the expression profile of the embryo. That's the first step. There is a second step, I think, that is partially genomic, in which we think that this canalized, the newly formed canalized circuit can be stabilized and completed by retrotransposal insertions, which are represented here by this colored symbols. So this insertion has essentially two roles. One role is to nail down the newly formed canalized pathway and stabilize them on the epigenetic embryonic landscape. And the second is to provide recognition sequence for the binding of regulatory factors, hormone receptors, promoters, splicing site, and so on. So in other words, they provide all the equipment that is necessary to circuit, the genomic circuit, to be functional. This is the same that what I just mentioned to you and described to you, but is in the context of an early embryonic development. Again, you know that I described this. It's the tissue that released the information that is taken up by the sperm. Now, this represents the landscape, the epigenetic landscape of the sperm, and this represents the epigenetic landscape of the oocyte. So when the sperm fertilized the oocyte, this event triggered the formation of a new embryonic pathway, genomic circuit. And of course, they are independent one from the other because they occur in the two different pronuclei. They are still independent when the two pronuclei come together in the same nucleus, and they are still independent at the two-cell stage because although the maternal and paternal genome are in the same nucleus, the genome are not fused. But they become fused at the fourth-cell stage, and in this case also at this stage also the newly formed pathways, the newly formed canalized pathway are overlapping, and they are become unified, forming the real embryonic pathways. Now, all this that I said is theoretical, so it's an hypothesis. So the real crucial question is how can this Waddington model of canalized landscape move from a metaphor to an experimentally testable hypothesis. And this is a real crucial question, and I need to try to explain how this could happen. I need to to open a parenthesis and briefly report some of the outcome that we obtained in another research program that was done in parallel with these embryonic studies, and was essentially focused on the role of line 1 in cancer. And we think that some of the outcome that we obtained from this work could provide some clues to answer this question. So briefly, years ago, we found that the reverse transcriptase inhibitors, those currently used in the treatment of AIDS, have a strong anti-cancer effect. So we studied this phenomenon, and we found that these inhibitors act as stressing agents on the cancer cell, and cause a number of variation, including the most important is a rearrangement of the chromatin. So, more specifically, we found that when cancer cells are exposed to these inhibitors, they induce, there is an upregulation of line 1. The upregulation of line 1 trigger an autophagic response in the cell. And then there is it's followed by a cascade of events that include a fragmentation of nuclear lamina with the rearrangement of the lamina-associated domain that they are important structure, functional and structure of the chromatin. There is an increase in DNA damage and increase of gamma to X. Most important, this promotes the newly formed interaction between the line 1 encoded proteins, both ORF1P and ORF2P, with both with lamin B1 and with gamma to X. There is a rearrangement of the peripheral heterochromatin, and there is, at the end, a reprogramming of the transcription profile. Now, all this effect are amazingly similar to what happened in embryo soon after fertilization. There is, in the embryo, there is an upregulation of the of line 1. All this occurs that there is an active autophagy. There is a full rearrangement of the interaction between the nuclear lamina and the genome, and the rearrangement of the lamina-associated domain. The DNA of the zygote, 1-cell stage and 2-cell stage, are heavily damaged, and there is a remodeling, quite substantial remodeling of chromatin. And it's known that there is a epigenetic reprogram of the transcription profile. So altogether, this data it's suggested that the sperm-delivered regulatory RNA could act as symmetry breaking agents, which are equivalent in the same role that the stressor have in somatic cell. Therefore, they induce epigenetic variation. And in particular, the combination of DNA damage and lamina fragmentation increase the plasticity of the chromatin and create favorable condition for the remodeling of the nuclear landscape. In this context, in the context of this remodulation, new genomic circuits can be formed. And these genomic circuits are not random event. I mean, the whole rearrangement of the chromatin probably is not random, but it's driven by the information contained in the regulatory RNA that is delivered by the sperm. And of course, this is from this a new transcription profile emerge. So on this ground, this is my last model, my last slide also. So this is a schematic representation of chromatin. This is the nuclear membrane, the nuclear lamina, the lamina-associated domain, and the chromatin loop. So after fertilization, the combination of DNA breaks and of lamina alteration and fragmentation in the context of an autophagic activation increase the chromatin plasticity that create favorable conditions for a controlled disassembling of the chromatin and induce a permissive variation-prone condition that can reshape the epigenetic landscape. You see, this is has a different shape than the starting one. And in this rearrangement, in this disassembling and rearrangement, the new genomic circuit can be formed. And again, I like to stress this, they are not random, but they are driven by the information contained in the RNA delivered, the regulatory RNA delivered by the sperm. So depending on the nature, the intensity, and the duration of the stimuli, stimuli that to which the somatic cells were exposed, and that they are reflected, the stimuli, on the composition of the sperm, on the sperm RNA. So this this could generate a variation that could be transmitted to the progeny, and it's likely that weak or mild stimuli would generate mild transient effect in the progeny. In contrast, the stronger or longer exposure and to the stimuli could generate permanently assimilated variation. And I would like to conclude saying that I think that this kind of mechanism could generate epigenetic variation for evolutionary novelties. Thank you.
Thank you, Corrado. That's kind of an extraordinary hypothesis there. Kind of takes them all in. Short question.
Why are you manipulating that? How do you imagine this is all happening in the setting of extensive protection against the impact of both the human RNA, human interacting RNAs, and so on? That is just happening in spite of those defense mechanisms.
Well, I think it's a natural component.
Can you go to my post?
Yeah, you can
Big question.
Huh?
He wants to know how this can happen in the presence of inhibitory RNAs.
If you expose the embryos to the inhibitory RNA, you stop the development. And as far as I can tell, in our experiment, the embryo was sensitive from the fertilization to the 8 cell stage. After the 8 cell stage, they are resistant to the treatment. So I think, and the same was if we inoculated antisense line 1 sequence, and you drop down the expression, again, you get an arrest of the embryonic development. So I think it's key.
That's an amazing mix. But I'm just wondering, how do these somatic RNAs or the exosomes, have you visualized them passing the blood testis barrier to get into the testes? How do they get through the blood testis barrier?
It's probably, this is all happening in the epidermis.
Okay.
He wanted, the question was, how do you anticipate that these somatic exosomes get to the sperm? And it's not going through the testis, it's going through the epidermis.
Because we know that, I mean, we purify the exosome from the sperm, and the RNA was in the sperm.
Okay.
Yeah. We have seen the exosome kind of aggregate of sperm. That's what we think. The circulation to get incorporate the content of the exosome into the sperm. That's our statement. But I have a question about the reverse transcriptase. The reverse transcriptase that is into the sperm, it works while the sperm is intact or extracellular once the content of this sperm side of it is in the sperm.
It is in the sperm.
Right.
We also have electron micrograph showing the spot of.
No, I'm fascinated by it, but the point is you have to release it. So once you get into the cytoplasm of an egg, for example, that reverse transcriptase start to work, or you work when the cell is intact. That's my question.
It works in the sperm, and it works independently from the sperm also in the early embryos. The early embryos are endowed with a reverse transcriptase.
So I told all activity in the sperm, other than the activity, a role kind of dormant under the sperm directly into the egg. That's what I mean. So there is also polymerase inside the sperm.
There is RNA polymerase, definitely. That was a very old finding.
That's what we're trying to CRISPR on those, and we need this enzyme.
So, one last question, is there anything about this model that doesn't agree with your work?
We haven't, in the system where we do look at Cree expression in the brain or in adipose tissue, we see the epididymis light up. With the exception of two Cree drivers, synapsin 1 and nested, we do not typically see any recombination in the next generation, which if most of the circulating Cree was going through the Spadafora pathway, we would expect a lot more red in the next generation. So while we do see red in the next generation for synapsin and nested, we don't see for most Cree producers, we don't see that, and I think that would be if all exosomes in the circulation willy-nilly made it into epididymal sperm and therefore into the kids, we would expect to see more red. But they're not sort of like, they're not mutually exclusive, but there may be some selectivity in which exosomes can do this.
Right, are there any other questions?
That was very provocative. So I have a question related to how many cells in the body can actually have this plant one active. Some time ago, I read that some cells have like a trigger zone, they have some line elements moving around. Is there any other cell other than the early embryo and the brain that you can have a cell active?
So he wants to, Jose is asking, what types of cells in the body normally have active line one? Use the microphone.
Brain.
It's active in the brain, and as far as I know, it's the only tissue somatic differentiated in which it's expressed, except then in embryos and in tumors, it's expressed.
And embryonic stem cells.
And brain.
Brain.
John
Embryonic stem cells.
Kennedy has reported that it increases as you age, or your ability to suppress line one. That's one of the, to point out the theories of the cancer. Like that's intriguing, because one of the things that stuck out in Oliver's talk is he mentioned that you never see cancer of the epididymis. And now you've got a fascinating observation of the epididymis handling line one differently. And perhaps that theory is
The epididymis has almost no mitochondrial cells. You make an epididymis, and that's it. And it is entirely testosterone dependent. So if you castrate a mouse, the epididymis disappears in what we, it's really fast.
Yeah, to be clear.
And it has two antibody and the epididymis for mitotic, so almost infinite. The only other thing I want to add to this is that years ago, we looked at all kinds of male reproductive tract tissues for endogenous retrovirus expression. And the epididymis is by far the highest level of expression, either virus particles or viral RNA is in the epididymis. It is silent in the testis and extremely active in the epididymis. Yes.
So is there any special trophism of these exosomes that will end up in the epididymis?
That's a question for Oliver. How do exosomes end up in the epididymis?
We don't know. The Cree expression and looking at the epididymis lighting up, the project sort of ground to a halt because at this point we sort of have two possible hypotheses. The exciting one is that the way the epididymis learns about the world is that the brain extracts salient features, compresses them into exosomes and sends them to the epididymis.
The postal delivery.
Yeah,
Right.
and the boring hypothesis is that the epididymis is a garbage can for exosomes, and Cree locks have infinite signal to noise, and so there's no physiological consequence under normal circumstances. The way that we could do this would be to stress an animal, look at some change in epididymal physiology, protein production, small RNA production, whatever, and then stress an animal, take his serum, dump it into a naive animal, and see if you reproduce the thing. So when we did that with trying to transfer Cree, we could transfer Cree, but it was in 6 out of 20 trials, and we were transferring like 20 animals worth of serum into the recipients. So we never figured out, and until we could make that more efficient, there was no way we were going to do things like fractionate serum for vesicular subpopulations, or do these like stress and transfer epidemiological physiology experiments. So that's sort of where we're stuck now. But that's how one would approach the question.
Okay, one more question.
Just a quick one. You suppose that it's just in mammals or what animal groups with this?
Is your model just for mammals?
It's the sperm, the appearance of the sperm to become
This permissive variation from conditions that you're expecting. I think we think it's across the animals.
Yes, it is. We tested the ability of the sperm to take up molecules from the echinoids to mammals, not all of them, but a number of them. And all of them have this ability. So I would say that this model could be in principle applied to different kind of species. It's not specific for mammals.
I'm guessing that happy hour is here. And we're going to end to have